When we look at the embryo and we look at the cervical region of the embryo, what we see on its outer surface is the series of ridges or bumps separated by a series of grooves. And those bumps are called the pharyngeal arches, and the grooves are called the pharyngeal grooves. Now, in order for us to really understand what's happening here, what we want to be able to do is look at the outer surface of the embryo and the inner surface of the embryo's throat at the same time. And so we'll make a section like that, remove part and look at what's left, and we'll be looking at something like this. So here's the outer surface of the embryo on one side, here's the outer surface of the embryo's neck on the other side, and then here's the interior of the throat with a pharynx. If we go upward in that direction, we're heading toward the mouth. If we go downward in the opposite direction, we're heading toward the esophagus. Since this is the outer surface of the embryo, we expect that it's going to be lined by ectoderm, and that's what it is. And the inner surface is, of course, lined by endoderm, the surface of the gut tube. And then in between the ectoderm on the outside and the endoderm on the inside, we would expect to see mesoderm filling in these regions here, and we do. But not only mesoderm. This is a part of the embryo to which a lot of neural crest cells migrate. So in between the ectoderm and the endoderm, this is filled with mesoderm and neural crest. So what we see is that the ectoderm on the outside has a series of invaginations, which are called pharyngeal grooves. On the inside, the endoderm has a series of evaginations called pharyngeal pouches. And between those pairs of grooves and pouches and the next groove and pouch, we have these bulges, which are called the pharyngeal arches, lined by ectoderm on the outside, endoderm on the inside, and filled with mesoderm and neural crest. Now, we have 5 pharyngeal arches in humans, but they're numbered 1, 2, 3, 4, and 6 because lower species have 6, and we know that the one we're missing is number 5. So we number it that way. And then the grooves and the pouches are numbered 1, 2, 3, and 4, and they're numbered according to the arch that's right in front of them. So if that's the first arch, that's the first groove, and that's the first pouch. Now, before we get into a detailed discussion of what happens to these in the human, it's helpful if we first have a little bit of an understanding of why we have these and how they function in fishes, since this is really just a throwback to the fish. So what happens in fish is that the pouch gets real deep at each level, the groove gets real deep at each level, and finally that endoderm of the pouch and that ectoderm of the groove fuse to form a double-layered membrane at each level, and then the membrane ruptures to create a slit at each level. And those slits are called gill slits, and these arches between the gill slits are called gills. And so what happens in fishes is as the fish swims in that direction, the mouth is opened, water comes into the mouth, down into the pharynx, and then passes right on back out into the ocean by going through those slits. And in passing through the slits, the water is being washed across the surfaces of these gill arches. There are lots of blood vessels right near the surface of the gill arches, so the blood that's in those blood vessels are able to extract oxygen from the water that is washing over the surface as it passes through those slits. So that's what happens in fish. In people, something almost the same as that happens with regard to the first groove and the first pouch. Almost the same. What happens in people is that the first groove gets deep, the first pouch gets deep, the ectoderm of the groove and the endoderm of the pouch come together and form a double-layered membrane. So that's exactly the same as fish. Where the difference comes in is that in fish that membrane ruptures, in people the membrane does not rupture. We retain that membrane and we call it the eardrum, or the tympanic membrane. So if we appreciate that the tympanic membrane is going to be a fusion of that ectoderm and that endoderm, that means that this groove is the region just outside or just lateral to that tympanic membrane, which means that that groove is going to be the external ear canal. It therefore also means that this pouch, which is the region just medial or internal to that membrane, that's going to become the middle ear cavity. We know that the middle ear cavity is continuous with the pharynx, and we can see that that pouch is continuous with the pharynx. Well, in us, the continuity is called the eustachian tube, or the auditory canal. So this becomes the external ear canal, this becomes the middle ear cavity and the eustachian tube. That, combined with that, becomes the tympanic membrane. Well, if this groove is what's going to become the ear canal, then this arch and this arch, which are forming the boundaries of that ear canal, must be the structures that form the boundary of the adult ear canal, and that's going to be the elements of the pinna, the external ear. So the pinna is formed from the first arch and the second arch. Now, when I say it's formed from the first arch and the second arch, remember that those arches are filled with mesoderm and neural crest. And as we'll see shortly, the neural crest cells that are in these arches will largely go on to become skeletal structures, either cartilages or bones or ligaments. So since the pinna is largely made up of cartilage, it's the neural crest of the first and second arch that contribute to the pinna. We know that inside the middle ear cavity, which is what forms over here, we know that inside that middle ear cavity we're going to have some bones. We just finished saying that neural crest of these arches is what's going to form skeletal structures such as bones. And so what happens is some of the neural crest of the first arch and some of the neural crest of the second arch migrates into this first pouch and ends up forming the bones of the middle ear. The neural crest of the first arch gives us the malleus and incus. The neural crest of the second arch gives us the stapes. We also know that in the middle ear cavity we're going to have two skeletal muscles, the tensor tympani and the stapedius muscle. Well, we've said that in these arches there's mesoderm, not just neural crest, but mesoderm. The mesoderm in these arches is going to go on to form skeletal muscles. So those two muscles that are in the middle ear cavity come from mesoderm of the first arch and mesoderm of the second arch. The first arch mesoderm gives us the tensor tympani. The second arch mesoderm gives us the stapedius. The reason this is useful for us to know is understood when we now add to this discussion the nerves that grow into each of these arches. As you recall, when we talked about limb buds, we talked about how mesoderm cells in the limb bud go on to form muscles, but that nerves grow into the limb bud to innervate those muscles. Well, the same thing happens here. We just said that there are mesoderm cells in these arches that will go on to form muscles. There are going to be nerves that grow into each of these arches to innervate those muscles. And what we're going to want to know is which nerve grows into which arch. And there's a specific cranial nerve or branch of a cranial nerve that is designated for each arch. So let me contribute that now. The nerve that grows into the first arch is the mandibular division of the fifth nerve. For the second arch, the nerve that grows into that is going to be the facial nerve. For the third arch, it's going to be the glossopharyngeal nerve. For the fourth arch, it's going to be the superior laryngeal branch of the vagus. And for the sixth arch, it's going to be the recurrent laryngeal nerve of the vagus. So coming back to the first and second arch, which we said the mesoderm of the first and second arch gives rise to the tensor tympani and to the stapedius. Well, now that we've said that the mandibular division of the fifth nerve is what innervates the muscles that develop in the first arch, that therefore will tell us that the mandibular division of the fifth nerve will innervate the tensor tympani that's in the middle ear. Because the seventh nerve innervates the mesodermal cells of the second arch, it will innervate the muscles derived from the second arch, which will include the stapedius that's in the middle ear. So the reason that the tensor tympani is innervated by mandibular 5 and the stapedius is innervated by the facial nerve is because those muscles are derived from mesoderm of the first arch and second arch, respectively. So let's now go a little bit further and see how this continues to develop on the next slide. On this slide, we now see that that first arch has gotten real deep. and it's in fact labeled the external auditory meatus, or the external ear canal. We see that the first pouch has gotten real deep, and it's in fact labeled as the middle ear cavity, and the eustachian tube, which we see is continuous with the pharynx. And the ectoderm and endoderm of those two linings are what become the tympanic membrane. That's what we've already said. And we said that this arch and this arch are going to form the boundaries of the canal, that is to say, the pinna. But notice that while on the previous slide we had a first groove and a second groove and a third groove and a fourth groove, on this slide we only have a first groove, and we don't see any other grooves. And the reason for that, I'm going to go back to the previous slide for a moment. The reason for that is what happens is that the second arch becomes very large and grows back and covers over all of this surface behind it. So this arch comes back, covers all of this over, and by covering it over, it ends up filling in those grooves. So we typically do not have any adult remnants of the second, third, and fourth grooves because the second arch has grown back and covered it over. And that's what we're seeing here. That's why we have this nice, smooth outer surface, which was the back growth of the second arch. In fact, if you recall, in the adult neck, there's a muscle that sort of comes back and covers everything else over, a very superficial muscle. That's the platysma muscle. And that platysma muscle, in fact, is the muscle derived from the mesoderm of the second arch, which came back and covered all of this over. So that's why it's a very superficial muscle. And because it's from the second arch, that's why the platysma is innervated by the facial nerve. Now, that's what happens normally. But sometimes, as the second arch comes back and covers this over, its job of filling in the second, third, and fourth groove is sometimes done only incompletely, and we may have a remnant of the second groove that's not completely filled in. And that remnant of the second groove that's not completely filled in is then called a lateral cervical or a pharyngeal cyst. So a pharyngeal or lateral cervical cyst is a remnant of a groove. It could be the second groove. It could be of the third groove, and then it would be found here. It could be of the fourth groove, and then it would be found here, or any combination thereof. And so since we know that these cysts are remnants of a groove, and since we know that the groove is lined by ectoderm, that means a lateral cervical cyst is lined by ectoderm. Okay, so that's an abnormality. Obviously, normally that doesn't happen, so we'll get rid of them now. Next, we have to look at the pouches. Now, we've already talked about what happens to the first pouch. We said it becomes a middle ear cavity in the eustachian tube. The second pouch is retained, and we still have an evagination in the wall of the pharynx, a little bit lower down than where the eustachian tube comes in, and that's called the tonsillar fossa. And so that's where the palatine tonsils are going to live. So the tonsillar material is forming just under that surface, but the epithelium lining the tonsillar fossa is from the endoderm of the second pouch. Now, the endoderm from the third and fourth pouches is a little bit more challenging to understand because those cells migrate. They don't stay where they are. They migrate. We know that the endoderm of the third pouch migrates caudally, like that. Some of it migrates for a short distance and then heads medially, and that's going to become the inferior parathyroid gland, IP. And some of it migrates much further caudally, goes all the way down into the chest, and that's going to become the thymus. Now, while it's true that the inferior parathyroid and the thymus come from the endoderm of the third arch, those endodermal cells are influenced or induced to become those structures under the influence of underlying neural crest. Remember that there's neural crest in here, and so the neural crest is inducing the endoderm of the third pouch to become the inferior parathyroid and the thymus. So without that neural crest, we'll have imperfections in the development of those structures, even though they are endodermal structures. For the endoderm of the fourth pouch, something similar happens. They migrate also. Those cells migrate also. They don't migrate as far. Some of them migrate pretty much straight medially and become the superior parathyroid gland. and some of them also migrate medially, and they become incorporated into the thyroid gland itself, which we'll talk about in a moment. Now, they are labeled here as UB, which means ultimobranchial body, but what's important is to know what they become. And those cells become the C cells, or the calcitonin-secreting cells of the thyroid gland. And that is, in fact, neural crest cells that do that. So we've now taken care of the pouches. We've taken care of the grooves. We haven't yet discussed how the thyroid gland itself gets there. And the thyroid gland forms as an evagination of the endodermal lining of the gut tube, but instead of it being a lateral evagination like these pouches are, it is a ventral midline evagination. Now, ventral midline evagination of the gut tube, that's a familiar theme. We've seen that several times already. That's how the respiratory diverticulum developed. That's how the hepatic diverticulum developed. That's how the ventral pancreatic diverticulum developed. In each case, it was a ventral midline evagination of the endoderm of the gut tube. So here we're seeing it one more time, a ventral midline evagination. It grows out from the ventral midline of the gut tube in the region of the floor of the pharynx, where the tongue is later going to develop. And that evagination, very much like the respiratory diverticulum and very much like the hepatic diverticulum, grows out, grows down, and then the distal end of it expands, and that distal expansion becomes the thyroid gland. That evagination is called the thyroglossal duct. Now, if that's where development ended, if that's where the story ended, the thyroid gland would now be an exocrine gland with a duct leading into the pharynx. But of course, that's not what happens. The thyroid gland is not an exocrine gland, because this thyroglossal duct, after the gland has expanded at its end and developed, then the thyroglossal duct fuses and disappears, leaving us with a thyroid gland down here in the neck that used to be communicating with the floor of the mouth and the region of the tongue, but no longer is. Okay, so we have this dotted line representing what had been the path of the thyroglossal duct, but normally that's not there anymore. Sometimes when that thyroglossal duct is supposed to fuse and disappear, now here's another familiar theme, when things are supposed to fuse, sometimes they don't do so perfectly, and there's a region that doesn't fuse. And so we may have a region where the duct doesn't fuse, even though it fused here and here, leaving us with a little cyst, and that is then called a thyroglossal cyst. A thyroglossal cyst, it's a remnant of the thyroglossal duct that should have fused but didn't, and so that'll be somewhere in the ventral midline of the neck. Thyroglossal cysts are lined by endoderm, since this is all an evagination of the ventral midline endoderm of the pharynx. So lateral cervical cysts that we talked about before are lined by ectoderm. Thyroglossal cysts are lined by endoderm. We said that this thyroglossal duct develops from the region of the floor of the pharynx where the tongue develops. And if we look at the floor of the mouth where the tongue develops, right on the tongue, there is a little pit in the floor, in the tongue, and that pit is called the foramen cecum. And the foramen cecum marks the site where the thyroglossal duct evaginated from. It's now a blind ending pit, so that's what cecum means, blind ending. But there is an abnormality in which sometimes the cells that should become the thyroid gland fail to descend, that the thyroglossal duct doesn't form. So these cells don't descend down into the neck, but they nonetheless become thyroid cells anyhow, and they remain right here on the tongue. And that would result in what's called a lingual thyroid gland. So a lingual thyroid gland, thyroid tissue forming on the tongue, is a result of cells of the thyroglossal duct not descending as they should. So now we've talked about the thyroglossal duct, we've talked about the pouches, and we've talked about the grooves or clefts, and that just leaves us with the arches themselves. We've already said that the arches contain mesoderm and neural crest. We've already said that the mesoderm largely becomes muscles, and we've said that the neural crest largely becomes skeletal structures, mostly ligaments and bones. We also said that into each arch a nerve will grow, a particular nerve. So indicated here is the nerve that grows into the arch. What the asterisk is there to remind us of is that this nerve does not develop from the arch like these structures develop from the cells of the arch, but rather the nerve grows into the arch. So what this is saying is that for the first arch, the mandibular division of 5 grows into it, and then it goes on to say that the mesoderm of the first arch develops into these muscles plus these muscles, and therefore the mandibular division of 5 must be innervating these muscles, and they are. The neural crest of the first arch is going to form the maxilla and the mandible. We'll talk about that in a few moments. And we've already said that it will also form the malleus and the incus, bones of the middle ear. The nerve that grows into the second arch is the facial nerve. Mesoderm in the second arch becomes a whole lot of muscles, the muscles of facial expression plus these 3. And therefore the facial nerve innervates all the muscles of facial expression plus these 3. And then we said that the neural crest will develop into skeletal structures. We've already said it develops into the stapes, and now we can add to that the styloid process and part of the hyoid bone. The nerve that grows into the third arch, we said, is the 9th nerve. The only muscle developed from the mesoderm of the third arch is the stylopharyngeus muscle. Therefore, the 9th nerve only innervates the stylopharyngeus muscle. The neural crest gives rise to a bit more of the hyoid bone. Some of the mesoderm in the third arch is retained to become a blood vessel. It's called the third aortic arch, which ends up developing into carotid arteries. The fourth arch, we said the superior laryngeal branch of the vagus grows into it. The muscles of the pharynx and one of the laryngeal muscles, the cricothyroid muscle, develops from that mesoderm. Therefore, this nerve innervates those muscles, and the neural crest gives us the thyroid cartilage. Some of the mesoderm becomes another aortic arch or artery, and that will end up being on the left side, the arch of the aorta, on the right side, the subclavian artery. And lastly, we said the recurrent laryngeal branch of 10 grows into the 6th arch. All of the remaining laryngeal muscles are the muscles derived from the mesoderm of the 6th arch. Therefore, this nerve innervates those muscles. And the cartilages, other than the thyroid cartilage, the other laryngeal cartilages come from the neural crest. And again, some of the mesoderm remains as the blood vessels, pulmonary and ductus arteriosus. So just coming back for a moment to that first arch, I want to emphasize that the neural crest of the first arch gives rise to the maxilla and the mandible, which means it's giving rise to a great deal of the structure of the face. So neural crest of the first arch is a major player in facial development. And when we think about maxilla and mandible, realize that means we're talking about palate formation, lip formation, tooth formation, all of which are related to the neural crest of the first arch. And since these are forming bilaterally, there's a right and a left arch, giving rise to right and left contributors to this facial development, all of this stuff needs to fuse together. And going back to that repeating theme, when things are supposed to fuse, sometimes they don't. So failures of fusion of these neural crest-derived structures can lead to cleft lips, cleft palates. Because teeth are forming in these maxillae and mandibles, they can lead to abnormalities of tooth formation if there are neural crest defects. So neural crest in the pharyngeal arches are really very important. And remember that neural crest is a cell type that migrates a lot, migrates to a lot of different places. And we've had this as a recurring theme that when there is one neural crest migration defect, there are apt to be other neural crest migration defects. So if we just review for a moment those structures that we have pretty well mentioned, with the exception of one or two things on this list. This goes back to that same list we looked at the very beginning of our conversation. The structures that we've talked about that come from neural crest include the adrenal medulla, include peripheral nervous system components that are seen in ganglia, sensory ganglia, and postganglionic cells in autonomic ganglia. I didn't previously mention it, but pigment cells, melanoblasts, come from neural crest. Schwann cells of the peripheral nervous system and some of the meninges. We just finished talking about pharyngeal arch cartilages and their important contributions to maxilla and mandible and face and pinna of the external ear and so forth. The odontoblasts for the teeth. We talked about the parafollicular cells that migrate into the thyroid gland to become those C cells. We previously talked about the aorticopulmonary septum and the endocardial cushion in heart development, all of that being neural crest. So now thinking back to the notion of neural crest migration defects suggests the possibility of other neural crest migration defects. And if we think about some of the major genetic syndromes that have multiple physical manifestations, for example, Down syndrome would be a very good example. And think of the many things that are associated, putting aside the issues of cognitive development. Putting that aside, think of some of the things that we see in Down syndrome. We see abnormalities of teeth, not at all uncommon. Missing teeth, misshapen teeth, that's neural crest. We see a much higher than average incidence of cleft lip and cleft palate. That's neural crest. A very high incidence of congenital heart disease associated with the aorticopulmonary septum and the endocardial cushion. Again, that's neural crest, and so on and so forth. I want to remind you that the postganglionic neurons that are found in autonomic ganglia include the terminal ganglia. Think about the terminal ganglia that are found in the wall of the colon. When there's a failure of neural crest migration into the wall of the colon, there's an absence of ganglion cells in the wall of the colon. That's Hirschsprung's disease. So that's another neural crest migration defect. So the common thread that ties together so many of these seemingly disparate physical abnormalities, the common thread is neural crest derivation of these structures and neural crest migration that didn't occur properly. So that concludes our discussion of the major elements of anatomy and development that are important for you to know for the USMLE. I urge you to focus primarily on the material that we discussed in these reviews. Those are the high-yield topics. It is not a good use of your time to be looking for other anatomy material that we did not discuss. That's not going to be high yield. That's not a good use of your time. If you review what we've talked about and really make sure you know it, I'm very confident that you'll do very well on the anatomy and embryology portions of the USMLE Step 1, and I want to wish you a great deal of luck in that endeavor.